I'm Suzanne Goh, and this is a recap of the presentation on mitochondrial dysfunction and autism spectrum disorders from the TACA National Conference in 2015. This is an image of an 8-week fetus in utero, and the process by which the brain develops into the mature adult brain is incredibly complex and intricate, and it's a disruption of this process which ultimately leads to the symptoms of autism. We now know an enormous amount about the disturbance to brain development that occurs in autism, and much of that is through brain imaging studies, and you can see the explosion of brain imaging studies of autism on this graph over the past 30 years. There are many different techniques to measure the function of the brain, the size, shape, and structure of different parts of the brain's chemical composition, and the connections between different parts of the brain. And from all of those studies, we know that autism is characterized by impairment in what's been called long-range structural and functional connectivity, so that the networks that connect very distant or long-range, far-reaching regions of the brain are relatively decreased, and instead we have an increase in local networks that connect nearby regions of the brain. And this leads to a state of disconnectedness and disorganization, which occurs in very early in development, and that research evidence points to disruptions in very specific mechanisms that lead to this disruption. And that is a process of impaired synaptic pruning, or a problem with the pruning away or reduction of the connections between neurons, and a problem in the process of autophagy, which is the process by which cells die. And so that leads to an overabundance of cells and the connections between them, and we know that this impacts function across social, language, motor, and sensory domains, which is why a comprehensive approach to the treatment of autism needs to address all of those different domains. This is a research study of all of the brain imaging studies of autism that have been done in the preceding 30 years, and what we found was that there is strong research evidence showing disruption to many different learning and memory systems in autism, including the learning of facts and events, the learning of skills and habits, the learning of emotional responses, and other learning and memory systems. So is the problem with brain connectivity in autism hardwired and permanent, or can it change? Well, we know that it can change. And I'd like to show you several different brain images just to give you a sense for how important brain biochemistry and cellular health and function, and in particular the function of mitochondria, are to brain function and development. So these are some images of the brain taken with magnetic resonance imaging, where we're able to look at slices in 2 dimensions of the brain. And here are a few examples of conditions in which brain development has been disrupted. This is a condition called tuberous sclerosis complex, and these regions that appear white in the brain tissue are parts of the brain where the cells of the brain, the neurons, have not migrated to the correct location during brain development. This is a condition, agenesis of the corpus callosum, where the large white matter fiber tract that connects the right and left sides of the brain that you see present here is missing. And this condition is also one in which autism, epilepsy, and intellectual disability is quite common. For most individuals with autism, there is no structural abnormality that can be detected on brain MRI. And that's very likely because the disturbance to brain function exists at the cellular microscopic biochemical level. Over time, an individual with autism may begin to show brain atrophy, which is a loss of brain tissue and an enlargement of the central areas of the brain that hold fluid. And that very likely, again, has to do with brain metabolism and biochemistry. And at the heart of that process are the mitochondria, which we'll talk much more about in just a moment. So in thinking about autism treatment, we know that there are neurological, biomedical, behavioral, and educational approaches, but all of them are really targeted at trying to improve brain biochemistry, neural networks, and brain function. So what are mitochondria? What is their role in autism? And what does that mean for treatment? I'd like to start with the human body and the different organs within the human body. And all of those organs, all the components of the human body are made up of cells, and you can see here an image of an actual cell, a microscopic image, and here a diagram that shows the cell and the structures within the cell. The body of an adult has close to 100 trillion cells. And here circled in red are images of the mitochondria, which exist within cells. Here you see only 2, but in an actual human cell you can have up to many thousands of mitochondria. Now, mitochondria are at the heart of metabolism, and metabolism can be thought of, in simple terms, as the process by which the body converts nutrients into energy. Mitochondria are sometimes called the powerhouse of the cell because they generate energy in the form of ATP. And the density of mitochondria varies from tissue to tissue depending on its energy requirement. So certain parts of the body, like the brain and muscles, have a very high energy requirement, and so they have a high concentration of mitochondria. This research article, which was published in 2010, was very important in drawing attention to the role of mitochondrial dysfunction in autism. And the researchers in this study found that 80% of children in the sample that they studied, 80% of the children with autism showed reduced mitochondrial respiratory chain complex 1 activity. So that is, complex 1 is a particular enzyme involved in the part of the mitochondria called the respiratory chain, and they were able to show biological markers indicating reduction or impairment in the activity of that enzyme. There have now been many other research studies also pointing to disruptions in mitochondrial function in children and adults with autism. This is a review paper in which the researchers looked at the results of 18 different studies on mitochondrial dysfunction and autism, and they found that 78% of those with autism had blood tests indicating mitochondrial dysfunction. And this is another research paper in which the researchers explain how mitochondrial dysfunction may potentially explain the range of different symptoms seen in autism, including symptoms in the gastrointestinal tract, endocrine system, and many others. Okay. I just would like to mention that mitochondrial dysfunction, the function of mitochondria is very complex. Mitochondria are the heart of many different essential metabolic functions. And there's some very interesting and important research being done at UCSD in the lab of Dr. Bob Navio, looking at one aspect of mitochondrial function, which is referred to as purinergic signaling. And several research studies have now been done in mice showing that this aspect of mitochondrial function is abnormal, and that there are certain pharmaceutical agents that target this and seem to reverse the symptoms of autism in mice, and now these are being studied in humans as well. Okay, I'll skip over some of these research studies and move on to this one. This is a research study that we published out of Columbia in 2014, where we used brain imaging to look for evidence of mitochondrial function, evidence of mitochondrial dysfunction directly in the brain tissue of children and adults with autism. And the technique that we used is shown over here on the left, called multiplanar chemical shift imaging. And using this technique, we were able to look at each cubic centimeter of brain tissue, and on average, the brain of an adult has about 1,400 cubic centimeters of tissue. So we looked at each one of those regions to see whether or not we could detect lactate, elevated levels of lactate, which is a biomarker of mitochondrial dysfunction. And what we found was that elevated lactate was present in 13% of those with autism and in 1% of controls, which is a statistically significant difference. And this image shows you an example in which we looked at this voxel of tissue here, and the spectra shows the presence of elevated lactate, and that's represented by the peak right here. We were then able to map out the distribution of lactate-positive voxels in our group of individuals with autism, and the red boxes show you the regions in which lactate was detected most frequently, and that corresponded to a part of the brain called the cingulate gyrus, which is very important for higher level integration of thought, emotion, and behavior. And so we think what we've identified is a region of the brain that is whose function is disrupted in some individuals with autism, and also the biochemical mechanism that has led to the disruption to brain function. This is a book that was published by Dr. Martha Herbert, who is a pediatric neurologist and a researcher at Harvard Medical School. It discusses many of the different medical complications that occur in autism. And she writes that problems with the mitochondria create problems for the whole body, in particular the brain, which uses huge amounts of energy. And she goes on to describe some of the triggers of mitochondrial dysfunction, which include gene mutations, shortages of key vitamins and minerals in the diet, certain chemicals, heavy metals, and drugs, certain bacteria and viruses, and stress. So one possibility is that mitochondrial dysfunction may be a final common pathway to autism, and that certain genetic factors, toxins, infection and inflammation, vitamin and mineral deficiencies, immune dysregulation, and drugs, that these may ultimately lead to the symptoms of autism because of their impact on mitochondria. So mitochondrial function is important for clinicians and parents to understand because it appears to be present in the majority of children with autism. It may be a final common pathway to the symptoms of autism, and it may be a treatable cause of autism. So how is mitochondrial dysfunction diagnosed? We start with some of the common symptoms of mitochondrial dysfunction. And because mitochondria are throughout the body and play such an important role in the function of all different systems of the body, there's a very wide range of symptoms that may result. These include developmental delay or regression, language impairment, social impairment, intellectual disability, a range of neuropsychiatric symptoms, seizures, headache, hearing impairment, weakness, exercise intolerance, small stature, gastrointestinal symptoms, endocrine disturbance, and many, many others. There are some classic symptoms that point to mitochondrial dysfunction in a particular patient, and these include developmental plateau or regression, especially in the setting of illness or other stressor, such as following a vaccine or after a surgical procedure, symptoms in more than one organ system, and family members with diseases that have been linked to mitochondrial dysfunction, of which there are many. And you can see them here. They include a variety of autoimmune conditions, neurodegenerative diseases, and metabolic disorders. There are many different potential tests for mitochondrial dysfunction, and this slide lists some of the first-line tests that are done commonly. There are certain blood and urine tests, potentially brain imaging. What's important to recognize is that many of these tests have low sensitivity and that testing may need to be repeated multiple times. Testing may need to be done under certain physiological conditions, such as fasting or during illness, and because of these challenges, a physician may recommend treatment even if the test results are negative. There are also many resources now that can aid a physician in quantifying the likelihood that an individual has mitochondrial dysfunction based on their symptoms, test results, and other factors. And this is just one example. There are also protocols that can guide physicians in testing. So this shows a flow diagram and the test that you might start with, what will be done next along the decision tree based on the initial results, different biochemical tests, and then different genetic tests, and potentially a skin or muscle biopsy. But that final step is very often not required. So how is mitochondrial dysfunction treated? I'd like to draw your attention to a very important practice guideline that was published by Dr. Richard Kelly at the Kennedy Krieger Institute, which is the Center for the Treatment and Management of Neurodevelopmental Disorders at Johns Hopkins University. And they have been treating mitochondrial dysfunction in children with autism for over 15 years and have seen consistent improvement and in some cases reversal of symptoms with the use of certain vitamins and supplements. And Dr. Kelly writes, our clinical experience at Kennedy Krieger Institute over the last 15 years has shown that a deficiency of mitochondrial complex 1 is a common cause of regressive autism. While permanent developmental losses can be substantial, recovery can be almost complete in some children when treatment is started early after the first episode of regression, and a partial response to metabolic therapy remains possible indefinitely. The mitochondrial cocktail that is used at Johns Hopkins includes these components: L-carnitine, coenzyme Q10, vitamin C, vitamin E, vitamin B5, and alpha-lipoic acid. And